Hi, so yeah, maybe I'll try to just use this one too, or should I?
Yeah, if you turn that one on.
Okay.
I'll turn it off.
Cool.
Otherwise, it'll be back.
All right. Is it working? Okay. Hi, so I'm Amanda Minnich from Lawrence Livermore National Lab. Thanks so much for having me. I'm here today to talk about the data-driven modeling pipeline that we've built for ATOM. So ATOM stands for Accelerating Therapeutics for Opportunities in Medicine. We're a public-private consortium, and our founding members are GlaxoSmithKline, which is a pharmaceuticals company, Lawrence Livermore National Lab, UCSF, and Frederick National Lab for Cancer Research, which is run by the National Cancer Institute. And we're working to transform the drug discovery process by bringing together the diverse biological data that pharma companies have with high-performance computing capabilities from the national labs and emerging experimental capabilities. So our approach is that we lead with computation and then support that computation with targeted experiments. And our goal is to have a data sharing setup so that we can build better models by getting data from a number of pharma partners. And what's really cool about ATOM and something that I really like working on is that we're building an open source framework. So we're going to release all of our code and as much of our models as we can out to the public. So this isn't a moneymaker for one particular company. So our current status is that we sit in a shared space in Mission Bay, San Francisco, and we have around 25 full-time employees engaged across the member organizations. Our R&D started in March 2018, so we've been working for about a year on the pipeline that I'm going to present, and we're in the process of constantly engaging new partners. So on the last slide, I'll have an email address. If you think your organization might be interested, you can email them. So the reason that we want to work on changing the drug discovery process is that it doesn't work very well. It takes a long time, it's really expensive, and at the end of this, on average, 6 years of development, there's only a 12% clinical success rate. So you're doing all of these experiments sequentially, and you have to wait until one drug fails before starting new experiments on the next drug, and it just seems very inefficient and ripe for innovation. So our ATOM workflow concept is to have fast in silico loops that are then supported by slower experimental loops in order to parallelize and speed up the process. And this is our overall molecular design platform. It's somewhat complicated, and I'm just going to be talking about the stuff in the red box. So the data-driven models that generate key safety and pharmacokinetic parameters that then get fed into higher order models and become part of the overall loop. So I'm going to quickly talk about the infrastructure that we're using at Livermore and then go through the pipeline that we've built and then the results of some experiments. And okay, I think I know what's happening. It's doing automated timings. That's why it keeps moving. So I'm going to just do, like, what is happening? All right. Hopefully, God. All right, I'm going to quick fix this because this is going to drive me bananas. Okay, well, we'll see. All right, so the infrastructure. We have a Docker Kubernetes cluster that we've set up. So all of our data scientists are working out of their own Kubernetes pods, and they have Jupyter Lab as a front end for their development environment. We also have all of our data services running out of individual pods. And then we have our HPC clusters that the labs are known for. They have a ton of resources for us to use, but they don't have any data services provided. So we've been able to use port exceptions to make REST API calls to our data services on our Kubernetes cluster. So data services are really important, I think, all the time, but especially for biological data. We need to be able to organize our raw data, also our curated data sets, our model-ready data sets, the train test validation split of our data sets for when we're building our models, the serialized models themselves and all associated metadata, the performance results when we generate predictions for these models, and then the outputs of simulations, because we're also doing simulations for this project. And these data types really vary in size, format, and level of organization and complexity. So we've built up a variety of services to handle these needs. The first is our data lake, which is basically an in-house object store service. So it allows for the association of all kinds of metadata with any kind of file. You can also just store the reference to a file if the data needs to stay on an encrypted drive, which is true of our pharma data, or if the files are really big. It has buckets for access control, and you can access it via a GUI or a REST API. And then we also use Mongo for a number of things for our data lake metadata, our model zoo metadata, and our results database as well. And then we have some MySQL pods because a lot of public data sets are available in that format. So our overall structure, we have all of our backend services deployed via containers, and then we have our server-side APIs that talk to our application client APIs, and then the two ways that the user accesses it are either through our HPC systems or through Jupyter Lab. All right, so this is the overall schema of a data-driven modeling pipeline, so I'm going to go through each piece and just describe how we've answered that part. So for data ingestion and curation, we got around 300 gigs of raw pharma data from GlaxoSmithKline on a variety of bioassay and animal toxicology data. So that corresponds to data on around 2 million compounds. The matrix is fairly sparse for assays and compounds, but we do have it covering almost 2 million compounds. And then domain experts from GSK used Jupyter notebooks to process the data. We found this worked really well because some of them were not as familiar with Python, so that's a nice intro. And also you have the markup language to write all the comments in, and the code itself, so you can rerun it and reproduce all of the processing steps, but also sort of read it to get a sense of what happened. And so we've curated around 150 model-ready data sets. So these are mainly for safety and pharmacokinetic assays. And we've generated descriptors. They're called mode descriptors. They're the kind that we've done so far. And they're basically float vectors that describe different chemical properties of the molecules. So we've done that for all of the GlaxoSmithKline data, and then also for Chembl, which is a public source of compound data, and also for enamine, which is around 680 million compounds. So this was something where our supercomputers helped, and we now have that available. So we support loading our data sets in either from the data lake or the file system. We understand that when we release this framework, other people aren't going to have the same data services. So we want to make sure that we support that at every step of our pipeline. We allow for, right now, extended connectivity fingerprints, which are like a vector of ones and zeros that describe the molecule, graph-based features, those mode descriptors, and now we've incorporated some open source descriptors as well, autoencoder-based features, and we've written it in a way that people can hopefully just plug in their featurizer class that they want to use to the pipeline to allow for that flexibility. And we added in a splitting of the data set based on structure. So normally you would want to do some kind of random split, but with pharma data, you have an initial compound and then you make little tweaks to it to try to optimize for whatever you're trying to treat. So it's very clumpy. So we split the data sets based on structure, and we're working on improved ways to do that as well so that our models are truly learning and not just memorizing. So we found, as is true in basically every aspect of data science, featurization is the most important thing. It varies by data set what works best, but those float descriptor vectors have outperformed ECFP, which is kind of a standard way of featurizing, and also our graph-based features. This is work that my colleague Kevin McLaughlin did. I just wanted to touch on some ways that we're trying to process these features. So we've used dimensionality reduction here, an algorithm called UMAP. It's like t-SNE, but it's faster. It's a manifold-based method. And what you do is you train a model up that will then reduce the dimensionality of your data, and you can weight the training of that model by the response variable if you'd like. And so here, the gray is no dimensionality reduction. Blue is dimensionality reduction, but no weighting. And here we weighted by the response value. And the y-axis is average precision. So as you might expect, if you take into account the thing you're trying to predict when you change the feature space, you're going to then do better. So we're working on different things like this to try to then process our feature vectors. We've built an overall framework to train, tune, and predict for our models. We currently support sklearn and then DeepChem models. DeepChem is an open source library out of Stanford, and our pipeline uses DeepChem a lot. It's specifically for molecular data, and it's a wrapper for TensorFlow, and it's a really good one. And then we also allow for custom model classes. We're working on incorporating PyTorch models as well. And yeah, we tune the models using the validation set and perform k-fold cross-validation. So we support distributed brute force hyperparameter search. We support linear grid, logistic grid, random, and user-specified steps. We don't support more advanced optimization techniques yet, but that's sort of next on our list to incorporate in. But basically, you specify your input with a JSON file. So here, the shortlist key is the file that lists all the data sets you want to build models for. And then I'm saying here I want to do neural net and random forest with 2 different kind of featurizers. The learning rate 0.0001 to 0.2 in 10 steps. And then these different layer parameters kind of get zipped together. And then it'll generate all combinations specific to the model types and send those out as jobs on our clusters. And then if you specify it, everything will get saved in our model tracker database, and then you can do analysis to pick out the best performing models and things like that. And so even with this sort of naive brute force approach, we have seen improved results with our hyperparameter search. So the x-axis is test set r squared, or the change in the test set r squared, or the change in the test set ROC AUC for classification. And we do find improvement, and I think that we can go further from there as we play more with the layer architectures and things like that. Okay. So our models are predicting binding, inhibition, and activation values for safety-relevant proteins, and also important pharmacokinetic parameters that we are then inputting into our QSP models. And we're working on some hybrid ML and molecular dynamics models. So when you run simulations, you get a lot of output, and we're working on seeing what of that output we can then input into our machine learning models. It hasn't been the most successful so far. There's so much output, but we're working on that. And also sort of the reverse, where you use machine learning to narrow down the number of molecules that you would need to do simulations for. We calculate model-based uncertainty quantification, and I'll talk about some of our results for that in a minute. And we support complex queries for model selection. So we save a huge amount of metadata with the model, because if we ever want these models to be trusted by the FDA, maybe someday, that these are valid, we need to be able to recreate every single step from raw data to prediction. So we save everything that we can think of with the model, and then we support queries. So you can say, I want a neural net model that was run between these dates that has an R-squared of higher than this on this data set with this feature type or whatever, in a JSON file or a dictionary, and it'll search the model zoo and load those up for you. We've been working a lot on different visualization modules as well. So this is an example that Kevin did, again, using UMAP, that dimensionality reduction technique. And I thought it was just a good example that he packed a lot of information into one plot. So the color is the error, and then the size is the actual response value. I think this is PIC50. And then the shape is the training, validation, or test set. So you can really analyze how your model is performing and potentially why. We're also doing chemical diversity analysis, and this is really important. So part of what Adam is doing is a de novo compound generation loop. So we're creating new potential drugs that we want to then generate predictions for. And so we need to have a sense of, is this something that the model should even be able to predict for? Does it look enough like the training set to be valid? So we're doing a lot of different things. This is, we did some hierarchical clustering based on Tanimoto distance, which is like Jacquard similarity between these ECFP vectors, and then plotted the distance matrix as a heat map. Okay. So we've done neural net and then random forest models so far on ECFP the mode descriptors, and graph convolutions. For neural nets, we varied the learning rate, the number of layers, the layer sizes, and the dropout rate. In random forest, we varied the max depth and the number of estimators, the number of trees. And we train iteratively up to n epochs. I've been doing 500, and then we pick the best model based on the validation set performance. And we save all of those. So this is experiment summary as of January. I think we've probably closer to maybe 10,000 models now, but more neural net than random forest because we're experimenting more with those hyperparameters in both regression and classification models. So we've been finding that we've been doing pretty well with classification, with the caveat that these are really unbalanced classes. So we're working on ways to make the classes more balanced so that we can really feel confident in these numbers. But most of our ROC AUCs are greater than 0.8. And the assay size range from like 200 to 9,000 compounds, so these can be really small data sets. And 23 of the 28 we found improvement with neural net. One had huge improvement, so we're looking into why that happened. But classification does tend to be a lot easier than regression, and that's definitely what we've found. For these assays, they range in size from 100, which that's very difficult, especially for a neural net to predict anything for 100 compounds, up to 124,000. So it really varies for that given assay. And about half of them show improvement for neural nets. And descriptors tend to do really well. But the R-squareds are from 0.1 to 0.7, so we definitely have room for improvement there. And one of the things that we noticed is it really depends on the number of compounds in the data set, which you would expect, but as we get larger data sets, we tend to do better. So we're looking at ways to increase the size of our data sets. So we're sourcing public data sets wherever we can. We're generating targeted experimental data, so this is in process and we're getting new results all the time. And then also looking at things like transfer learning. So you train on a larger data set that's predicting a similar property and then tune the last couple layers on your smaller data set. Sorry, this is really annoying that it keeps moving. And then also multitask learning, so you predict on a bunch of related assays together. Okay. So for our UQ analysis, this helps to show us what the model isn't confident about, and we want to use that to characterize confidence in model predictions. So when we're predicting properties for molecules that we don't have ground truth for, we want to be able to use the UQ as a proxy for accuracy to guide our active learning and also to weight model ensembles. So for random forest, what we've ended up doing is just calculating the standard deviation of the predictions from the individual trees. We've tried fancier things, but this has seemed to work better. And for neural networks, we're right now just out of the box using DeepChem's method. And this combines aleatoric, which is like sensing uncertainty, and epistemic, which is the model uncertainty. So the way that they do aleatoric is they modify the loss function. So during training, you predict the response variable and the input variance, both. And then epistemic, during prediction, they apply a number of dropout masks and then look at the variability in the prediction value, and then they combine them. So our goal is to quantify prediction uncertainty for assays like this, and you'll immediately see these vertical lines, and this is one reason why regression is so hard. These are called censored values, and basically it means that if the value was less than something, they just put less than something, which is really bad when you're trying to do regression, and the model has a hard time with that. So something that's a little like cheering is that you see at the bottom where it's more accurate, it does have lower uncertainty values. Oh yeah, so X is actual, Y is predicted, and the color is the UQ value. So, and then as it goes up and it's less accurate, it has a higher value, but it still doesn't look like a really clear pattern. So we looked at the correlation, and it is indeed pretty low. So we binned the error so that we got rid of some of the real outliers and calculated Pearson's correlation between the UQ and error, and it ranges from 0.14 to 0.35. So there are different hyperparameters that you can actually tune for uncertainty quantification. So we're going to start working on those and also looking at other, like maybe Bayesian methods and things like that. And so another thing that we thought we would look at to see how well our UQ is doing is what if we set a really low UQ threshold? Would we be able to identify really low error samples? So we say experimental error is acceptable in our predictions, which is around 0.3 on a log scale. And we say if we set the UQ threshold really low, can we find these low error things over here? So we did precision recall curves varying the UQ threshold to look at that. And we found that the neural net doesn't reach a precision of 1 for any UQ threshold. So even if you set it really low, you're never going to get only low error samples. So it definitely shows us that we have work to do with this piece. Okay, so our current status, we have internally a Dev 1.0 release of our pipeline, and it's installable with pip as a wheel file, and it runs internally at GlaxoSmithKline for evaluation, so they're trying it out on some of their internal data that we don't have. And then our models have been incorporated into that de novo compound generation loop that I mentioned, and we have been able to export and share models with our consortium members as well. So our future plans, we want to release the pipeline open source. We've begun that process at least at Livermore, but since it is a multi-member consortium, there are multiple permissions that need to happen. And we want to Dockerize the entire pipeline and also release our data services infrastructure as individual Kubernetes pods, because particularly the model tracker and the data lake, I think, could be really useful for other people. And in terms of improving performance, we want to add in that optimized hyperparameter search function and explore those hyperparameters for uncertainty quantification. And then, as I mentioned, the transfer and multitask learning and starting to work with ensemble modeling. So if you have any interest or want to learn more, you can email info@atomscience.org, or if you have technical questions on the talk, feel free to email me. Thank you.
Amanda. Questions about Atom?
You mean to go? All right. So when you get all these drug I'm here.
Okay, thank you.
when you get these drug prospects, then what is the process within your consortium? Is it distributed among all of the members, or publicly shared, or what happens with the prospects?
So we're going to have a proof of concept session. Like, we haven't actually picked a target that we're trying to do this for yet, but in the third year we're planning to pick a target and work on that. And I think that information will be released because it's showing that our concept works, but then within the individual members, they can take the code and the tools and do whatever kind of compound generation themselves, and that's separate from Atom. So we're really focused on the tools and that piece, and not generating potential new drugs necessarily, other than for proof of concept.
I see. All right, thanks.
Mm-hmm.
And you're facing a couple challenges, both on probably stochastic responses as well as hyperparameter responses, right? And one of the big challenges with random forests is, by their very nature, they're stochastic. You're going to get slightly different answers every time you run them because of the random nature. And that's one of the reasons we focused on the iterative random forest, is it actually gets you stability. The answers converge, and then they're reproducible. Mm-hmm. And then that makes that a mineable space on the interpretability side that you just don't have with random forests by themselves. So
that's a very good point. Yeah.
We'd be happy to talk more about that and give some insight.
Yeah, that would be great.
The hyperparameters discussion on deep learning is a challenging one, right? And part of that, and Frank alluded to this, a lot of the existing neural network architectures are around ImageNet, right? They're driving, those rules of thumbs are recognizing cats and puppies. And those are not particularly good representations of scientific problems. And so one of the things that we are doing on Titan and Summit now is different sorts of hyperparameter sweeps with ways to actually change the neural network architectures and let the architectures evolve to fit the problem. And when you do that, you come up with architectures that you would never have thought of out of the box by tweaking the knobs for 6 months, which is what we all do for a while. Mm-hmm. And so this is a supercomputing solution to hyperparameter hyperparameter space. And we're finding this a useful way to approach these sorts of problems as well as simulation models, that you can view your parameter space as an X matrix and your model output as a Y, and all of a sudden you're under a problem that explainable AI can approach as well. So there's sort of some interesting intersections we can do to help solve those sorts of problems. And I mean, we've bruised our foreheads on very similar problems for a long time, and once you have a subdural hematoma or two, you start finding solutions.
Cool, that's great. I'd love to talk more with you about that.
Yeah, great presentation, and good for you for being able to stay with it with that thing flipping forward. I'd never been able to keep talking if that had been happening. So good for you. But I'm just curious you hit on tracking the model for the FDA. I mean, the inputs and the way the model changes over time is going to be really important for not just drugs, but a lot of things. I'm just curious, how is that done? What's the state of the art today?
For
tracking and keeping track of what influenced the model, and just for fraud detection and things like that. If our lives are going to be run by neural networks, we need to know what came in to influence the neural network and what the states were at various points in time. I'm just curious, you as a scientist, where do you see that evolving?
It's an area of very active research right now, because we're trying to bring neural networks into cars and medicine and all of these areas. So we haven't on this project done any piece of explainability, but there's a lot of things with attention networks and people trying to peer in and look at the layers and incorporate that as you're training, like the fact that you're going to need to understand it as you're training. And I think that we will probably need to bring that into this at some point, but yeah.
You need the mic. I'm sorry.
Just in response to that, which is a great point, there's a fine line between understanding what the algorithm is doing at different layers of the network and explainability, interpretability of the mechanistic understanding of the science. Mm-hmm. And that it's an easy thing for that answer to bifurcate. And there's value in both in understanding what's going on within the algorithm and how the algorithm is performing at different layers. But that's a different answer from what's the feature engineered space that's responsible for your classification. They're both interesting problems. But they're somewhat discrete. And we sort of raster back and forth and get lots of questions sort of mixing those two concepts. And they are a bit distinct. And they're both important, but they're different.